Okay, so this talk was finishing the CAMI session. However, we are continuing with talks on the evaluation of metagenomic software. And now is Ziye Wang from Fudan University in China, assessment of metagenomic assemblers based on hybrid reads of real and simulated metagenomic sequences.
Thank you. Good afternoon, everyone. I'm Ziye from Fudan University in China. Here, I will introduce a strategy for evaluating the metagenomic assemblers, which is based on the hybrid reads of real and simulated metagenomic sequences. Let's go into the background part. Microbes are quite ubiquitous and quite important. Some people may even believe that microbes run the world. I think it's a good belief to do metagenomic research. And assembly is usually an essential first step for the follow-up studies in metagenomic research. Understanding the power and the limitation of the metagenomic assemblers is quite important for us to choose the right assemblers for further study. And the current computational experimental designs for evaluating the metagenomic assemblers can be divided into two main categories. The first category of studies evaluate the metagenomic assemblers using real metagenomic dataset. Without known reference, it's difficult to evaluate the integrity and accuracy of the assemblers. The other category of studies evaluate the metagenomic assemblers based on the simulated dataset. Since real metagenomes are generally complex and the microbial compositions are usually unknown, so the performance of the assemblers on the simulated dataset may not reflect that on the real dataset. And even if the simulated dataset is complex, some biologists like our co-author Furman, who still don't think it is real data, who still want to know how the assemblers perform on the real data. So we wondered if we could mix the simulation rates of the known species with the real metagenomic data sets and evaluate the assemblers. For this reason, we designed a strategy and do some experiments to show that our strategy is necessary to a certain degree. Here is the figure shows the framework of our evaluation strategy. Is quite simple. First, using the complete sequences of the selected genomes, we applied Mycin to generate the simulated reads. And after mixing the simulated reads with the real metagenomic reads, we assembled the hybrid reads with different assemblers. And taking the selected genomes as the references, we applied MetaQuest to obtain the statistics of the kinetics and for subsequent analysis. We selected 4 advanced metagenomic assemblers: Metaspace, MegaHit, IDBAUD, and FARSeq for evaluation. Our experiments show the advantages and weaknesses of the assemblers in terms of integrity, contiguity, and accuracy with different variables. The different variables include sequencing depth of the simulated data and the genetic difference between the simulated data and the real metagenomic data. The data set. One of the real metagenome dataset is a small-scale human gut dataset sample, and the other is a large-scale ocean sample. For these 2 real metagenomes, different NCBI genomes were selected for simulation. And for the human gut dataset, we chose different microbiome genomes from oral, freshwater, and wetland. And for the ocean dataset, we selected different genomes from ocean, different ocean, or the different location of the oceans. And what's more, to evaluate the performance of the assemblers on the strain value, we also selected 5 equivalent strains to be added into the human gut data set as the simulated sequences. The results part. We did some pre-experiments to see whether the selected genomes are contained in the real metagenomes and show the necessity of using a hybrid approach. To show the limitation of using the simulated metagenomic data only, we assembled the hybrid reads of the simulated data and the real data and compared the assembly with that based on the simulation data only. The performance of the per simulation is better, which meets our hypothesis. And to show the limitation of using the real metagenomic data, we assembled the reads from the genomes to be added into the human gut dataset and evaluated the assembly with using MetaQuest without providing reference genomes. If reference genomes are not provided for the MetaQuest, it will select the genomes with high genome fractions as the reference. We compared the assembly accuracy based on the MetaQuest recruit genomes with that based on the true reference. And the comparison shows that based on the MetaQuest recruit genomes, the numbers of misassemblies and mismatches are quite much higher than that based on the true reference genomes. We can see that take the misassembly as an example, is from 0 to 18. And the table shows the assembly integrity of different assemblers. The color from purple to red represents the result from good to bad. We can see that Metaspace has best performance in integrity, and Farseed perform worst in integrity at low sequencing depths. This page shows past results of the ocean data. The NGA50 is to measure the contiguity of the assembly. And NGA50 is similar to N50, but if a contig has a misassembly, the contig will be broken into smaller pieces. So I think it's more reliable. We can see that at the low sequencing depth, the NGA50 selectors of the assemblies from all the assemblers are relatively low, and the Metaspace has obvious advantages. This page shows past results of the assemblers on the strain level. We can see, just like before, the color from purple to red means the results from good to bad. We can see that Metaspace perform best on the whole, and Farseed perform is good in accuracy, but the performance of recovering the genomes is the worst. In conclusion, Metaspace perform best in integrity and contiguity on the species and level, and megahit follows. Farseed perform best in accuracy, but at a cost of low integrity and contiguity, especially at low sequencing depths. Megahit has the highest genome fractions on the strain level, and Metaspace has best overall performance. What's more, megahit is the most computationally efficient in our experiments. It runs really fast. Thank my advisors and co-authors. The full paper is published in briefing in bioinformatics. Thank you for your attention.
Time for a quick question. There are no questions, so I think in the interest of time, okay, we move to the next. So let's thank again Ziye.